Welcome to the deep dive. If you're looking for a shortcut to understanding, well, one of the most revolutionary advances in modern genetics, you are definitely in the right place. Today we are taking a deep dive into CRISPR-Cas technology. We're talking about a discovery so fundamental, so powerful, that people often compare it to the innovation of PCR, the polymerase chain reaction.
Exactly, and that's a huge comparison to make.
It really is. I mean, in genetics, progress is usually step-by-step, incremental.
Yeah.
But every so often, something comes along that's just a massive leap forward. CRISPR-Cas is absolutely one of those. It shed light on a totally new idea, adaptive immunity in bacteria, and at the same time, gave us this incredible tool for editing genomes. Efficient, cheap, revolutionary.
So that's basically our mission for you today. We're going to walk you through this step by step, looking at the biology, how it works, how it became a tool, and of course the ethical side. We're sticking strictly to the source material you gave us. The goal is to really clarify why this tech is seen as such a huge disruption, why it's such a big deal, and why it all started with bacteria fighting off viruses.
Right, let's start there. The origin story.
Yeah.
It's basically constant biological warfare between bacteria and the viruses that infect them, bacteriophages. We already knew bacteria had defenses, like restriction enzymes. Those are kind of like innate defenses, right? General protection.
Yeah, innate immunity is sort of a blunt instrument, a general defense. But CRISPR-Cas, that discovery turned things upside down. It showed that bacteria, which everyone thought were too simple, actually have adaptive immunity.
Like our immune system, learning from past infections.
Exactly like that. A mechanism that learns, evolves, gets better at fighting off things it's seen before. It builds a memory. Nobody thought bacteria could do that.
So how did they figure this out? The structure came first, didn't it? Back in '87.
That's right. Researchers found these strange regions in bacterial DNA, clustered, regularly interspaced short palindromic repeats, CRISPR.
Pagey name.
Yeah. But the structure was puzzling. You had these identical repeated DNA sequences over and over.
But separated by these unique non-repetitive bits called spacer sequences.
And those spacers were the mystery for a long time. Until around 2005, that was the big aha moment. 3 different research groups working independently showed that those spacers, they were actually bits of DNA from bacteriophages, viruses.
Whoa. So the bacteria were literally keeping snippets of their enemy's DNA.
Precisely. It was like a molecular most wanted list, a memory bank of past attacks stored right there in the genome.
Okay, that's wild. But how did they prove it actually worked as a defense? The Danisco story.
Ah, yes, the yogurt company, Danisco. They were just trying to make their Streptococcus thermophilus cultures resistant to phages that could ruin yogurt and cheese production. Purely practical goal.
So what happened?
They exposed their bacteria to a specific phage, the ones that survived. Well, they became resistant, but only to that exact phage strain.
Specificity. Okay.
And here's the kicker. Those resistant bacteria had acquired new spacers in their CRISPR region, and those new spacers perfectly matched the DNA of the phage they'd just fought off.
Wow. Direct evidence.
Absolutely. And they tested it further. If they deleted those newly acquired spacers, the bacteria lost their resistance, poof, gone.
Okay.
But if they artificially inserted those specific viral spacers into bacteria that hadn't seen the phage before, those bacteria became resistant.
Incredible. So it's specific, it's adaptive, they learn, and it actually stops the virus.
Textbook coevolution. Bacteria acquiring memory in real time to fight back. Amazing stuff.
Okay, so that's the why. Now let's get into the how, the machinery. You mentioned the CRISPR loci and these Cas genes nearby.
Right. The Cas genes code for the Cas proteins, the enzymes that do the work, like cut in DNA or RNA. We're focusing here on the type 2 system from Streptococcus pyogenes, the bug that causes strep throat.
Why that one specifically?
Because it's relatively simple. It mainly relies on just one key enzyme, the Cas9 nuclease. That simplicity is why it became such a powerful tool later on.
Makes sense.
Yeah.
So how does this Cas9 system work in the bacterium? What are the steps?
Okay, think of it in 3 main stages. First is spacer acquisition. When new viral DNA invades, Cas proteins, specifically Cas1 and Cas2, seem to be involved here. Chop up that invader DNA into little pieces called protospacers. Then they insert one of these protospacers into the CRISPR array, usually right at one end, near a leader sequence, adding it to the memory bank.
Okay, memory acquired. Step 2.
Step 2, cRNA biogenesis. The cell needs to make use of that stored memory. The whole CRISPR locus, repeats and spacers, gets transcribed into one long RNA molecule, the pre-cRNA.
Just a long string of RNA copies of the repeats and spacers.
Exactly. But for the type 2 system, there's another player, a separate non-coding RNA called tracrRNA. This tracrRNA actually base pairs with the repeat sections of that long pre-cRNA.
Okay, so they stick together? Then what?
Then Cas9 binds to that RNA duplex, the pre-cRNA, stuck to the tracrRNA. And then another enzyme, RNase 3, comes along and chops this whole complex up into individual mature units. Each unit is a short cRNA derived from a spacer, still bound to a piece of the tracrRNA.
So each mature unit has a unique spacer sequence telling it what virus to look for.
You got it. That crRNA duplex is the guide. It's loaded onto the Cas9 enzyme. This whole complex now patrols the cell.
Looking for trouble, which brings us to step 3.
Step 3, target interference. The cRNA part of the duplex acts like a homing beacon. It scans the DNA in the cell, looking for a sequence that perfectly matches its spacer sequence.
And when it finds a match, say, on an invading virus's DNA.
Cas9 cuts it. Game over for the virus. But there's a crucial detail here. How does Cas9 avoid cutting the bacterium's own CRISPR locus, which obviously contains the exact same spacer sequence?
Ah, right. That would be bad. Self-destruction. How does it avoid that?
This is maybe the most elegant part. It relies on something called the PAM sequence, Protospacer Adjacent Motif.
CAM. Okay. What's that?
It's a very short, specific DNA sequence, usually just 3 nucleotides, like 5-ORNG3 in the S-pyogene system, that must be located immediately next to the target sequence on the invading DNA.
So the guide RNA finds the matching sequence, and Cas9 checks if the PAM sequence is right next door.
Exactly. Cas9 will only bind stably and cut the DNA if both the target sequence match and the PAM sequence are present.
And the clever part is?
The clever part is that the CRISPR locus itself, where the spacers are stored in the bacterial genome, doesn't have these PAM sequences next to the spacers.
Ah, so the viral DNA has the target sequence and the PAM, but the bacterium's own copy only has the target sequence.
Precisely. That's how Cas9 tells self from non-self. It's a built-in safety check. So when it finds the match plus the PAM on the invader DNA, Cas9 uses its 2 nuclease domains, they're called HNH and RhoVC, to cut both strands of the DNA. A clean double-strand break.
Okay, that's incredibly neat. A programmable defense system with a built-in safety catch. You can see why researchers got excited.
Oh, yeah. That relative simplicity, Cas9 plus that dual RNA guide, got people like Marafini and Sontheimer thinking, could we use this outside bacteria? Could we program it?
And then came the 2012 breakthrough from the Doudna and Charpentier labs.
That was the turning point for making it a tool. They showed, in vitro, in a test tube, that you could simplify the system even further. Using genetic engineering, they fused the essential parts of the crRNA, the targeting sequence, and the tracrRNA, the part that binds Cas9, into one single molecule.
The single guide RNA, or sgRNA.
Exactly, the sgRNA. Now, instead of needing Cas9 plus 2 separate RNA molecules, you just need Cas9, and this one, easy-to-design sgRNA.
And you could design the sgRNA to target basically any DNA sequence you want, as long as
it's next to the correct PAM sequence. Suddenly you had a 2-component system Cas9 protein and a custom sgRNA that could be programmed to cut almost anywhere.
That sounds much simpler than the older gene editing methods like zinc fingers or talons.
Oh, vastly simpler and cheaper and faster. With ZFNs and talons, targeting a new gene meant complex, difficult protein engineering to make the DNA binding part recognize the new sequence. With CRISPR-Cas9, you just need to synthesize a new sgRNA molecule with the right 20 nucleotide targeting sequence. It relies on simple RNA-DNA base pairing, which is easy to predict and design.
Okay, so the potential was clear, but moving it from a test tube into actual living cells, especially complex ones like ours, that was the next hurdle, right?
Right, and that came quickly. In 2013, independent work from Feng Zhang's lab and George Church's lab showed they could get CRISPR-Cas9 to work inside cultured mammalian cells.
How did they do that? Just inject the Cas9 and sgRNA?
Essentially, yeah, but packaged correctly. They usually put the gene for Cas9 often tweaked with a nuclear localization signal, an NLS, to make sure the protein goes into the nucleus where the DNA is, and the sequence for the specific sgRNA onto plasmids. Then they introduce these plasmids into the cells.
And inside the cell nucleus, the sgRNA guides Cas9 to the target DNA, Cas9 cuts, and then what? The cell has a broken chromosome.
Right. And the cell hates having broken DNA. It immediately tries to repair it using its own natural repair pathways. And this is where the editing part really happens. The cell has 2 main ways to fix a double-strand break.
Okay, what are they?
The first is called NHEJ, Nonhomologous End-Joining. This is the cell's quick and dirty fix. It basically just jams the two broken ends back together.
Doesn't sound very precise.
It's not. NHEJ is very error-prone. It often introduces small insertions or deletions called indels right at the break site. These indels can cause a frame shift mutation, messing up the gene's code, and often resulting in a non-functional protein. So if your goal is just to knock out a gene, disrupting it, NHEJ is actually quite useful.
Okay, so NHEJ for gene disruption. What's the other pathway? The precise one.
That's HDR, or Homology-Directed Repair. This pathway is much more accurate. It uses an undamaged DNA template to repair the break correctly. Normally, it uses the sister chromatid or the homologous chromosome as the template.
Yeah. So it copies the information from the good copy.
Exactly. But researchers realized they could hijack this process. If you introduce not just Cas9 and the sgRNA, but also an artificial piece of DNA, a donor template that has homology to the regions around the break but contains the desired chain.
You trick the cell into using your template for the repair.
Precisely. The HDR machinery uses the donor template, incorporating your specific edits, maybe correcting a mutation, inserting a new sequence, or making a precise deletion. This is how you achieve precise gene editing.
That's clever. But HDR is less common than NHEJ, isn't it?
It is, generally. So researchers developed tricks to favor HDR. One common one is to engineer Cas9 itself. By mutating one of its cutting domains, say the Revisi domain, you create a nickase.
A nickase. It only cuts one strand.
Right. A single-strand nick, not a full double-strand break. The cell is less likely to repair a nick using the error-prone NHEJ pathway, which increases the relative frequency of the desired precise HDR pathway, especially if you use two nickases guided to nearby sites on opposite strands.
Okay, that makes sense. Boosting the chances of the good repair. But there's still the worry about cutting in the wrong place, right? Off-target effects.
That is the major hurdle, the infidelity issue. Cas9 isn't perfectly specific. It can sometimes tolerate a few mismatches between the sgRNA and the DNA sequence, especially away from the PAM-adjacent end of the target.
And you mentioned earlier this might have been useful for bacteria fighting rapidly changing viruses.
Exactly. A little bit of flexibility might help catch mutated viral sequences, but when you take Cas9 and ask it to find one specific 20 base pair site within the 3 billion base pairs of the human genome.
Which is what, 1,800 times bigger than the bacterium's genome?
Roughly, yeah. That inherent slight sloppiness becomes a huge safety concern. Cutting the wrong gene could have disastrous consequences, like causing cancer. Improving specificity and reducing off-target cuts is probably the biggest area of ongoing research for therapeutic uses.
Definitely a critical point. But even with that challenge, the versatility of the system is incredible. You mentioned using it for basic research, too.
Absolutely. One brilliant innovation was creating dead Cas9, or dCas9.
Dead. How does that work?
They mutate both of Cas9's cutting domains, HNH and RhoVC. So now dCas9 can still be guided to a specific DNA sequence by the sgRNA, and it binds there, but it cannot cut the DNA.
It just sits there. What's the use of that?
But you can attach other functional proteins to dCas9. Think of dCas9 as a programmable delivery system that can take almost anything to a specific spot on the genome.
Like what?
Well, you could fuse it to a transcriptional activator domain to turn a specific gene on, or fuse it to a repressor domain to turn a gene off. This is huge for studying gene function without permanently changing the DNA sequence.
So you can control gene expression precisely. What else?
You could attach enzymes that modify epigenetics, like histone deacylases, to study how those modifications affect gene activity at specific locations, or attach a fluorescent protein, like GFP, to dCas9.
To light up specific genes inside a living cell.
Exactly. Visualize chromosome dynamics or gene location in real time under a microscope. The dCas9 toolkit is incredibly versatile for basic research. It's a cornerstone of modern reverse genetics.
Amazing. And what about biotechnology? Beyond basic research.
Huge impact there, too, especially in agriculture and livestock. Faster, cheaper, more precise genetic modification.
Like the pig example.
Right. Porcine reproductive and respiratory syndrome virus, PRRSV, costs the pork industry billions. Researchers knew the virus used a specific part of the pig's CD163 receptor protein to infect cells. Using CRISPR-Cas9, they precisely edited the gene to remove just the small bit coding for that viral interaction domain. The resulting pigs still have a functional CD163 receptor, but they're resistant to the virus. Targeted, precise improvement.
And crocs, too.
Yep. DuPont Pioneer used CRISPR to modify the promoter region, the control switch of a gene called RGS8 in corn. They tweaked it to increase the gene's expression under drought conditions. The result: A drought-tolerant corn variety that apparently yields about 5 more bushels per acre when water is scarce. Again, precision targeting for a desired trait.
Okay, so research, biotech, now the big one. Medicine, gene therapy.
That's the ultimate goal for many, yes. Using CRISPR-Cas to directly correct the mutations that cause genetic diseases in patients.
How would that work? Say for a recessive disorder where you need 2 bad copies of a gene.
For recessive disorders, the ideal strategy would be to use HDR. You deliver Cas9 and sgRNA targeting the mutated gene, and a donor template containing the correct wild-type sequence. The goal is to correct at least one of the 2 faulty alleles in enough cells to restore function.
And for dominant disorders where just 1 bad copy causes the disease, sometimes the mutant protein actively causes harm.
Right. For many dominant, negative, or gain-of-function disorders, you might not want to just correct it, but actually inactivate the mutant alleles specifically. If there's a unique sequence in the mutant allele, even a single nucleotide difference, you could design an allele-specific sgRNA that guides Cas9 to cut only the disease-causing copy, leaving the healthy copy untouched. Then NHEJ would likely disrupt the mutant gene, effectively silencing it.
That requires incredible precision. Are we seeing progress in models?
Definitely. There's been promising work in mouse models for Duchenne muscular dystrophy, DMD. Injecting CRISPR components into fertilized eggs led to mice where the DMD mutation was corrected in some muscle cells. Even though the mice were mosaic, meaning not all cells were edited, they showed significant improvement in muscle function. That's very encouraging.
But editing embryos is one thing. Treating an existing person, like a child or adult with DMD, that's harder, right? Getting the CRISPR system into all the muscle cells.
Delivery is a huge challenge, absolutely. But there are strategies being developed. One called CRISPR-Gold uses tiny gold nanoparticles coated with the Cas9 protein and the sgRNA, plus a DNA donor template. They showed this CRISPR-Gold system could be delivered intravenously in mice and successfully edit muscle cells, leading to functional improvements in adult DMD mouse models. Still early days, but promising for in vivo therapy.
What about other diseases? Sickle cell.
Yes, sickle cell anemia is a major target. It's caused by a single point mutation in the beta-globin gene, HBB. Studies have shown you can take bone marrow stem cells from patients, or mice, edit them ex vivo outside the body using CRISPR-Cas9 and an HDR donor template to correct the HBB mutation.
Then put the corrected stem cells back into the patient.
Exactly. In mouse studies, these edited stem cells successfully engrafted back into the bone marrow and were still producing healthy red blood cells containing wild-type beta-globin months later. That's a potential cure pathway.
And this isn't just theoretical anymore. Clinical trials are happening.
They are. Particularly notable are trials that started in China, treating patients with aggressive non-small cell lung cancer. The strategy there is a form of cancer immunotherapy. They take T cells, the patient's own immune cells, out of the body. Then they use CRISPR-Cas9 to knock out the gene for PD-1.
PD-1, that's a checkpoint inhibitor target, right? It normally dampens the immune response.
Exactly. PD-1 acts like a brake on T cells. By knocking it out using CRISPR, the idea is to remove the brakes, creating hyper-aggressive T cells. These modified T cells are then expanded in number and infused back into the patient to hopefully attack the cancer more effectively.
Fascinating. Moving from the lab bench right into clinical trials, which inevitably brings us to the legal and ethical side of things.
It got complicated fast.
The patent battle, for instance. That sounds intense.
It was, and still is to some extent. It largely pitted Jennifer Doudna's group at UC Berkeley against Feng Zhang's group at the Broad Institute, my Harvard. Doudna Berkeley filed patents early, covering the general use of CRISPR-Cas9 as a gene editing system, especially based on their foundational in vitro work with the sgRNA.
But Zhang Broad filed later, specifically focusing on the use of the system in eukaryotic cells, like human cells, which they demonstrated first.
Right. The core legal question became, was successfully adapting the system from bacteriophages tubes to work effectively inside the complex environment of a eukaryotic cell an obvious step based on the earlier work, or was it a separate inventive leap deserving its own patent?
How did it shake out?
It's complex and involved multiple rulings and appeals. Generally, Berkeley holds foundational patents on the system components and basic usage, while Broad holds key patents on the use within eukaryotic cells, which is critical for most therapeutic applications. It highlights how valuable and contested this technology became almost overnight.
And beyond patents, the ethical debates are huge, especially the distinction between editing regular body cells versus germ-line cells.
That's the crucial ethical line, yes. Somatic cell editing targets non-reproductive cells in an individual. Any changes made are therapeutic for that person only and cannot be passed on to their children. There's broad, though not universal, agreement that this is ethically acceptable for treating serious diseases, provided it's safe and effective.
But germline editing, that's different.
Very different. This involves editing sperm, eggs, or embryos. Any changes made would be incorporated into every cell of the resulting person, and crucially, it would be heritable, passed down through generations.
And that raises all sorts of concerns. Unforeseen long-term effects, changing the human gene pool, enhancement versus therapy.
Exactly. We've already seen controversial studies where researchers in China and later in the US use CRISPR to edit non-viable human embryos, often targeting genes like HBB for beta thalassemia or MYBPC3 for a heart condition. These were explicitly done as proof-of-concept experiments to see if it could work and what the off-target effects look like, not to create babies. But they sparked intense debate.
So what's the current consensus, if there is one?
A major report by the U.S. National Academies of Sciences and Medicine provided some guidance. They supported moving forward cautiously with clinical trials for somatic cell gene therapy for serious conditions when there are no other reasonable treatment options.
But for germline editing?
They urged extreme caution. They strongly advised against using it for enhancement purposes, trying to make better humans. However, they notably did not recommend a complete ban on heritable germline editing for therapeutic purposes.
Really? Why not?
They acknowledged that for some couples, both carrying recessive mutations or one carrying a dominant one, germline editing might someday be the only way to ensure they could have biologically related children who are healthy and free from a devastating genetic disease. They left the door open, but stressed the need for much more research on safety and efficacy and broad societal consensus before ever proceeding.
So the ethical boundaries are directly tied to the technical capabilities and safety.
Absolutely. The risk of off-target mutations is concerning in somatic therapy, but it's existentially frightening when those errors could become permanent features of the human gene pool.
Okay, let's try to wrap this up. We've gone from a bacterial immune system
Discovered through weird DNA repeats and phage DNA,
to understanding the Cas9 RNA guide mechanism with its PAM safety check,
simplified into the sgRNA tool.
and then applied to edit genes in human cells using NHEJ or HDR repair.
Leading to breakthroughs in research, biotech like virus-resistant pigs and drought-resistant corn.
And now, clinical trials for diseases like cancer and sickle-cell, alongside huge patent battles and profound ethical questions.
It really is an incredible story, leveraging an ancient defense system into something revolutionary. Simple components Cas9 and an sgRNA, but capable of so much.
So, looking ahead, what's the final thought for our listeners?
Well, while people say the primary limit now might just be the researcher's imagination, the hard reality is still safety, especially off-target effects in our huge genome. So the question we should leave you with is this. Given those risks, particularly for changes that could echo down generations, what level of precision, what degree of certainty about safety and fidelity does science need to reach before germline editing crosses the line from technically possible to ethically responsible?
That really is the multi-billion-dollar and multi-generational question, isn't it? Something to keep thinking about. Thank you for sharing your source material with us, allowing us to take this deep dive into CRISPR-Cas with you today.